Hello, welcome to The Blueprint of Life. I am your host, Florence Peel, and if you clicked on this video today, you're in for a good one. I sit down with Dr. Emiliano Trucchi. He is a geneticist from Italy, and he works with species you could not possibly even imagine. It's very interesting work, and I hope you enjoy this conversation.
Okay, so my name is Emiliano Trucchi and I'm from Italy. I originally from Rome, but now I have a position in Ancona, which is an unknown, pretty much unknown city. Not that pretty, but nice overall, on the east coast of Italy. And my main field of investigation is, can we say, it's evolutionary genomics.
How did you get into this degree? Did you study biology and then led you to evolution? How did this process happen?
Yeah, actually, the getting closer to genetics and then genomics was not as straightforward as you can imagine. So I started as an ecologist, actually more in the entomology. So I was actually studying the ecology of insects after wildfire. That was my master thesis. And then, I did an internship. After the master thesis, I didn't get the PhD immediately, so I waited a little bit. And then I got an internship in an ancient DNA lab. So there was the early times of ancient genetics, ancient human genetics. And there was this group in Rome. I was in Rome during my master and PhD in two different universities. And this group was actually the only one doing ancient genetics in Italy, ancient DNA. And so I joined the group for an internship. I liked it, and I got closer to all the molecular lab and the analysis, genetics. And then I decided, okay, I want to do a PhD in this field. And then I planned an experiment, a study on gorilla, just based on a few samples which were available in that lab back then, but then not realizing that to study gorilla you need much broader network or collaboration. So it actually was just a project I wrote for the PhD application, which was good because I got the PhD fellowship, but then I had to change project. And my PhD study was on the Italian porcupine. So in Europe, in Italy actually, there is this species of porcupine which are actually represented in Africa and India, for example, but not in the rest of Europe. So the thesis was, my study was to assess whether this porcupine population in Italy was native or introduced in ancient times. And we discovered that the population was introduced during the ancient Roman times, towards the end of the Roman Empire, probably. And
to track that exact kind of time period there.
Yeah, because using the molecular clock, pretty much we got samples from North Africa, we counted the differences and said, okay, all these differences between these two lineages can be dated back about 1,000 years ago, and that is the end of the Roman time.
Wow.
So that was my first molecular study, and then I took I did some experiences abroad. So after the PhD, I left Italy for Norway, and I got this European fellowship, which is called Marie Curie Fellowship. Is a very prestigious one. Back then, it was very nice to get it, and I went to Oslo, and there I joined a big group of ecological and evolutionary synthesis. It was a very nice group with lots of nice people from all over from all over Europe and the States. So it was a very good environment, and there I started genomics there. So they had lots of money, they could approach genomics, and that was the time where we were actually transitioning from genetics to genomics, because this was the beginning of the 2011, 2012, there was this next generation sequencing revolution. So it was possible to actually do genomics on any non-model species, any species. And that was the time I started with that. And after that, out, I stayed 3 years in Norway, and then I got another postdoc in Vienna for 3 more years, and that was more on plants. So I moved from animals, because in Norway I continued on the porcupine, and I started my most important collaboration because I was sharing the room with another Marie Curie fellow, Céline Leboeck, and she was actually leading a monitoring program on penguins in Antarctic and subantarctic islands. And there we joined forces, and she didn't have anyone doing genetics or genomics of penguins, and I said, okay, yes, let's do it. This is an empty niche, and I started with the penguins there. And then the penguins kept going in my research, in my investigation. So they are still the main part of my research. So this time in Vienna, after that, was kind of an intermediate phase. I was on plants, and we were approaching other part of genetics like epigenetics, small RNA, transcriptomics, and I learned a lot. So with plants, even if I was never a big fan of working with plants in general. Animals are funnier.
Yeah, more interesting for sure.
Right?
Yeah.
But of course, you can do lots also with plants, so it was nice. And then after these 3 years, we decided it was about time to go back to Italy and to bring back what I learned abroad. And I joined a group in Ferrara, which is a city north of Bologna, doing mostly conservation genetics, conservation genomics. And that was I went there. They were working on the Apennine bear. That was the topic of the first seminar. And then after I got a little grant there to study penguins again, and then I got the position where I am now in Ancona, where I started my group. So now I'm an associate professor in Ancona, and my group is called Genomics Lab, and and we are actually we need someone like you doing the old social media manager and things. So
I'll send this on to you so you can
thank you.
100%.
But I like you being here and going around and taking everything and transforming into nice news.
It's nice news.
Very nice job.
I agree. I really like this. Yeah. So it's still very interesting, your trajectory here. I mean, it's not really normal for geneticists to work on the species that you work on, to not have a model species, but actually work with natural animals. Was that something that you always knew you wanted to do, or was it just how the work unfolded?
I think both. So the things just unfolded this way, but I don't know. I always made this point with when I'm asked about why I'm not working with model species, because with model species you have more resources, you can do more things, you have bigger network, papers get more citations and so on. But I still think that the approach towards model species actually is kind of narrowing down a bit what we can do. So the natural experiments out there with all of these different species that are out there, any of them can be a model of something, and we just need to pick the right species for the right question. So sometimes the question comes after, looking at some data set that you have for some other reason, and then you start getting acquainted with a certain species, and then you explore more and you get questions, and then you can answer, as was the case with the Apennine bears. Some other times, some species are already so special, like the emperor penguin is which is one of my favorite cases. It's the most cold adapted vertebrate, warm-blooded on Earth. So it's something that, evolutionarily speaking, is quite difficult to make. There are not many examples of that, right? So it must be special. So getting to know what are the genetics, the genomics underlying that special evolutionary lineage is something that I like to study, I like to know.
I really like that's quite a cool way, because it sounds like you approach everything with the first question of how do you evolutionarily exist? And then we're going to start unpacking that. That seems like a really wide question going there. It's the question here, take your time to answer it there, but I'm curious, you've had so many different model species and so many interesting findings. What has been one of the most pivotal, incredible moments you've had in your research experience?
That's a very difficult question. because I'm always
I
using.
think you have a lot.
No. Actually, research, and when I discuss with others, we always agreed on that. That actually, if you choose to just give your life to research, you need to be prepared to an ocean of frustration, which sometimes is actually pointing towards little moments. You have nice islands of happiness and this surprise moment, just to fall again into frustration soon after. This is what science is, and there are lots of small moments where I think I realized, okay, we are actually seeing something. We are getting closer to something that is actually, I don't know, sometimes I'm so happy to see something that should be there, because there are models saying that selection exists, for example. And once you see that actually you have some evidence of selection, some signature of that, I'm so surprised all the time, even if it's something that it's there, but that you can really touch it. And I would say that so I think if I go on describing some of these little moments, that would be really too technical. Sometimes I'm really happy about really so technical things, and people won't be that interested in that. But maybe
Well, I don't know. I think you explained well.
So, for example, a recent one that I remember was that 2-3 years ago, I was actually analyzing in a different way some data of penguins, genomics data of penguins, and then I was just sorting synonymous and non-synonymous sites. So there are synonymous where the variance is changing, they are not changing the protein, non-synonymous and the protein is changed. And then I realized that there was a weird sorting of these variants according to the expression level of the genes they were in. So I found these variants in different genes, and then I realized that the way that they were actually present in the genes was somehow related with the expression level of the gene. And I was so surprised. This is incredible. And then I found that maybe there was a late 90s, someone described this evolution anti-correlation pattern, which is something that exists, so that it was seen in differentiation between species, that the accumulation of deleterious or non-synonymous mutation actually is inversely correlated with the gene expression. So the more a gene is expressed, the lowest the amount of changes it actually accept. So, and this was known, and then I could prove it in the standard segregating in the standing segregating variation, which was really cool in the penguins, and using the penguins as a case study. So before it was known for Drosophila or the model species, and then it was a very neat demonstration using any natural population like the king and emperor penguins. And this was something that took a while to get out, because it was really into maths and modeling and lots of theory. But then finally was published on July, May, June this year.
Wow.
in molecular biology and evolution. So it was a nice catch.
Oh, this is so cool. And now with this inverse relationship that you're seeing so well modeled on the graph with the penguins, what does this mean for your work? What does this tell you?
Yeah, it means that, for example, if we look at genes which are not that expressed, so they are used maybe only in some developmental stages, or only when they are needed, we don't know when, during the whole life cycle of a penguin, for example, then these genes can accumulate more variants, and they could host. So more variants means more evolvability, right? So the pace of evolution depends on the variance that is there. So genes with more variants can be the key for evolutionary changes. The genes that are highly expressed, maybe are housekeeping genes that are more constrained and tend to be preserved as they are. And so you can actually consider the accumulation of variation in a different way, and can decide, okay, maybe these genes that are not so expressed can be more important for changes. They can hold the key for the future adaptations. Instead, some others are less variable, and if you see some mutations there, maybe these mutations are more likely deleterious, so you can rank your mutations according to where they occur. Highly or lowly expressed genes, for example.
Wow,
No, this is super cool because as we are probably seeing in your work so much, like animals must be evolving continuously because of human impact and things like that are next to it. This might be a bit of a weird question, but since you started with an ecology background, I can imagine you're someone who's very environmentally conscious of human impact. Is that true? Would you say about yourself?
Yeah, I would say so. So I'm I'm aware that I'm paying. Yeah, that nature pays a toll because of us.
Yeah.
Right.
Great.
Yeah, I know that. I'm also an evolutionary, so ecology by formation, but evolutionary biologist as something. So in my perspective, I would say. So I think that nature will survive us and will persist no matter maybe in a different shape, in a different combinations of biodiversity. And if we want to just save nature as it is now, or if we want to work in conservation, for example, we do it for ourselves. We do it because we like it. So it's a very human way of conserving. So I think there is a human perspective in conservation, which is important. We need to be honest about that, right? And so the other thing is also that evolutionary times are so long and things change in such a long time scale that whatever happens today is actually pretty much nothing compared, right? So yes, I'm aware. I would love to preserve and to get more pristine habitats back, but I also
You're also an evolutionary, like researcher.
Things go on. Yeah, things go on.
Yeah, I just find that kind of interesting. Like that is a great combination of the two different perspectives there.
Yeah.
Super cool. And so I guess my last question here is, so please take it on. You've done already work with porcupines, with penguins, with the talk about the bears are super interesting. All of this crazy things about mitochondrial changes, genes with tameness. I thought that was crazy. What do you want to do next? Like, what is something you're really excited about?
Yeah, so I have a couple of grants that I've submitted waiting for approval, and actually I would like to focus more on aging pathways, but more as aging as related to, again, not just something that is relevant for humans and from a medical perspective, but can be maybe in the future, but more aging as related to evolutionary longevity. So how different species can evolve different lifespans. So what is the main drive? What are the drivers there? Because we, in the models that we know, in the paradigm is that everything. So aging exists because late after your reproductive peak, there will be no selection if you age or if you go bad. If your body just decays, expires. Exactly.
Yeah.
So the selective pressure for longevity cannot be at the end of life because there is no selection acting. So selection can only be effective before the reproductive peak. So how different lifespans can evolve in different species. So where is the pressure to get there? And my hypothesis, and this is the main thing I'm trying to get money to prove it, is that the evolutionary lifespan is actually a byproduct of some more streamlined or some selective pressures acting early in life. So to get a good body, all of the cells in the right place, good metabolism, and everything which is acting at the beginning of life stage of life before the reproductive peak, that is fine-tuned by selection. And whatever changes in that particular part of life is changed by selection. And the lifespan evolves because this fine-tuning happening early life by chance or just by coincidence is also increasing lifespan. So aging, not aging, could be a byproduct of a better development from the very beginning of cell differentiation to the reproductive maturity, let's say.
So you want to test this critical window and see how are you going to do this without a model species?
With the penguins.
With the penguins as
yeah,
well?
because they are long-lived.
Oh,
and we can use that.
But how are you going to get penguins that had like a very brutal upbringing and penguins that had a very good one? I don't know how you do.
Yeah, the idea is to get is to get thousands of penguins sequenced, which we have by chance.
Fantastic.
And just do a GWAS, so genome-wide association studies for penguins, for individuals that got to reproductive maturity, surviving till there, and individuals that didn't. To 5 years, they got to reproductive maturity at 5 years. And we say, okay, if we have two groups, control, so survivors and dead, and we make the comparison. And we do the GWAS. And then my expectation is that there will be some targets, some genetic targets showing up. Let's say 100 or so. And then we're going to follow these traits to check whether they are more represented in individuals living longer. So we will sequence 1,000 more individuals getting to 15 years old and 1,000 individuals getting to 25 years old, which we have available, and see if the difference accumulated here is the same as the difference accumulated down here at the end of life. So this is the overall idea that I got.
Wow.
I need some money to do that. Not much though, 1 million or so.
Because you already have the
Because thanks to this great collaboration with this group in France, they are following and getting samples from the penguins since 2000. So it's 25 years of sampling. They have more than 20,000 birds, blood sampled, and with a radio tag, so they know where they are, if they are coming back, if they are successfully breeding, how long they live, and so on. So we have lots of life history data that we can match with the genomics.
Super fantastic.
That would be great. So it's difficult. Again, as you said, it's difficult to make the funding agency believe that you can test this kind of stuff. Not in a model species.
But you prove that you can. It's such a
I think you can
way of doing that.
Because we need to show that we are. So this hypothesis is actually saying that the evolutionary lifespan depends on ecological pressure or real selective pressures at the beginning of life. Mm-hmm. We cannot do this in a lab because there's no selection in a lab. Or you cannot apply artificial selection. It needs to be a real selection acting on these groups at the beginning of life and then continuing to exist towards the end of life. So this is the only way. So this kind of analysis, this kind of test, you can only do in a real case scenario. So cannot be in a lab, cannot be on a mouse or Drosophila or Arabidopsis, the model species.
Oh, so fantastic! You're totally towing this really interesting line between natural and like medical type of experiment. That's very cool.
I'm trying.
Super nice. Okay, this is perfect. This was a great conversation.
Thank you.
Hello again. So if you enjoyed this conversation and would like to watch more videos like this, you can follow us at the Blueprint of Life Instagram, Facebook, YouTube, Blue Sky, and check out our website. Thanks so much.